Hi everyone, I'm Dr. Sean Elias from the Jenner Institute, University of Oxford, and today I'm going to be talking you through how scientists develop and test a new vaccine from start to finish.
Firstly, what is a vaccine and how does it work? Vaccines work by presenting antigens, molecular markers found on disease-causing agents, to the immune system. In response, specific components of our immune system, antibody-producing B cells and T cells, are formed that recognise and remember these antigens. Then, if the actual disease shows up with the same antigens, the immune system can quickly respond to kill the disease and protect against infection. There are two common types of disease we need vaccines for: endemic diseases, such as malaria and tuberculosis, and outbreak diseases, such as Ebola and, more recently, COVID-19. For many endemic diseases, we have existing vaccines, but they may only be partially effective. So, we often look to new technology to try and improve on them. For outbreak diseases, it is often very hard to predict when a new disease might emerge, so development of a new vaccine is often reactionary. However, we can often learn from previous outbreaks of similar diseases. For example, for COVID-19, we already knew that the spike protein on the outside of the virus was a good antigen target for a vaccine from work on other outbreak coronaviruses, such as MERS. So what are our design options for the vaccine once we know the disease we want to work on? The simplest type of vaccine is made using the disease of interest directly, either killed or in a live but disabled form, known as a live attenuated vaccine. This approach often delivers numerous antigens directly to the body, but can be quite reactogenic, meaning it can cause numerous side effects. Alternatively, you can just use the antigen of interest alone, an approach known as a subunit vaccine. Sometimes, the antigen alone doesn't produce a strong immune response, but we can add something called an adjuvant to the vaccine to help encourage the immune system. Another approach uses the genetic code for the antigen of interest, rather than the antigen itself. We use this code to construct vaccines, which in turn instruct cells of the human body to produce the antigen. The COVID-19 vaccine we have in development at the University of Oxford is a variant of this approach. In our approach, we have taken the genetic sequence of the spike protein and inserted it into a common cold virus that infects chimpanzees, an adenovirus called ChAdOx1. When this virus infects human cells, it encourages them to make the spike protein, which the body then produces an immune response against. The cold virus itself has been modified, so it cannot naturally replicate in human cells or cause disease. Adenovirus vaccines are currently also in development for other diseases like HIV, malaria, and Ebola, and have been tested on tens of thousands of people. As such, they are considered a universally safe approach for vaccination. The Oxford COVID-19 vaccine is nonetheless undergoing extensive clinical trials to test how safe and effective it is.
In the following video clips, I'll take you through the various steps in the process, from the initial production of the vaccine seed stock for use in humans, through to how we test the immune system in the lab. So sit back and enjoy.
The E1 region of the ChAdOx1 genome encodes for the proteins necessary to initiate virus replication. In our vaccines, we replace this region with our vaccine antigen, which means that the virus will no longer replicate in cells within the host's body. However, to produce ChAdOx1 vaccine stocks, the proteins expressed from the E1 region are required. To do this, we use a special cell line called human embryonic kidney 293 Treg cells, which express the E1 proteins. The virus can replicate successfully in these cells, and in doing so, kills the cells it grows in. We can observe this as cytopathic effect, or CPE, under the microscope. As more virus grows, we transfer it to fresh cells in more flasks to keep increasing the amounts of virus stocks. This method is good for making small stocks of vaccine, but for bigger batches, you need large vats of cells in suspension, rather than flasks, to make enough virus. So how do we separate our virus from the cells in the hyperflasks? Free virus that has already killed its host cells will be found in the flask supernatant and can be poured off for collection. However, there will be some remaining both in or attached to the cells. To remove the cells from the flask, we use an enzyme called trypsin to detach the cells. The flasks are then washed and cells plus remaining supernatant collected. We then spin these in a centrifuge to separate the supernatant containing the virus and pellet the cells. Supernatant can be used to infect more cells or continue down the purification pathway for making the final vaccine product. So we don't waste any virus, we also resuspend the cell pellet and freeze thaw the cells to break open any remaining cells and release leftover virus. Our clinical grade vaccine is manufactured in clean rooms following the principles of GMP, Good Manufacturing Process. This is to ensure it is safe for use in humans in our vaccine trials. After filling and crimping our vaccine vials, each undergoes a rigorous quality control process. We check that the vials have the correct volumes and that none contain any impurities. Approved vials are finally labelled and then transferred for storage in our freezers, ready for use in our clinical trials. Volunteers taking part in our clinical trials attend appointments at the CCVTM. Once they have been successfully screened to ensure eligibility for any given trial, they can be booked in to be vaccinated. In our COVID-19 vaccine trial, 50% of participants receive the test vaccine and 50% receive an alternative control vaccine. Which vaccine is injected is blinded for both participants and those administering the vaccine. Volunteers are monitored post-vaccination to check for side effects and assess the overall safety profile of the vaccine. Before their first vaccination and at subsequent visits over the preceding weeks and months, volunteers donate blood so that we can assess the immune response to the vaccine. The teams of scientists responsible for this job are based both at the Oxford Vaccine Group at the CCVTM and the Jenner Institute at the Old Road Campus Research Building. Blood samples are collected from clinical trial volunteers in special tubes called vacutainers. There are different coloured tubes for different ways of treating the blood. Green tubes contain anticoagulants that prevent the blood from clotting, whilst red tubes allow the blood to clot naturally. Volunteer blood samples are anonymised. However, each blood tube has a unique barcode that contains information about that participant, including their unique ID number and the time point at which the blood sample was collected. This allows us to track the samples throughout the trial and make sure that all samples have been received in the lab. Once we know the right samples are in for the day, we can start preparing the tubes. These are the tubes we use for the various processes that allow us to separate and store the blood. The first step is to isolate serum from the red top tubes. Serum is a straw-coloured liquid that contains antibodies which can be used to look at the immune response to vaccination. Serum is typically frozen down and then used in later assays, such as the ELISA. Unclotted blood from green top tubes is poured into special tubes which have a filter. This filter allows separation of the different components of whole blood. This process is achieved by spinning the blood samples at high speeds in a centrifuge. The special liquid in the bottom of the tubes allows for separation of the different components by density. As the most dense, red blood cells sink to the bottom of the tube below the filter. Next comes the clear-coloured liquid. On top of this sits a layer of cells, seen by the white band. Above this, on the top, is the plasma. This plasma is removed off the top and frozen down for future use. Plasma is quite similar to serum, but contains some additional components. With the plasma removed, we can then pour off the cells into a separate tube. We then add some media to the cells. This media has been pre-warmed to a temperature at which the cells are most happy. We have two types of media, one for washing and one for storing the cells up until the point where they're ready for the next step, counting. To count the cells, we take a small aliquot of cells and put them into some special tubes with a known volume. We then transfer these tubes to a special cell counter. We always count cells in duplicate to make sure that the counts are correct. Once counted, we can then adjust the cells to a standardized concentration. Once at the concentration that we require, we can use these cells in our special assays. These include ELISpot and flow cytometry. These allow us to look at the T cells within that population of cells, which give us an indication of how well the vaccine is working. The final step in the process is to freeze the cells down. We freeze the cells down using a special freeze mix. When put in the freezer, this freeze mix allows the cells to cool at a steady rate, preventing them from bursting and the cells dying. frozen cells can be revived to use in different assays in the future or repeat assays from the first round.
So when we make a vaccine, we're interested in two types of immune response, an antibody response or a T-cell response. And an ELISpot is a test to count the number of T-cells in your body that the vaccine has made your immune system produce. So we take a sample of blood, we separate out the T-cells from that sample of blood, and then we incubate them overnight with some small pieces, some small fragments of the vaccine. And if the vaccine's worked and your immune system has responded, when those T-cells see that part of the vaccine again, they'll interact with it and produce some chemical signals that we can detect on the ELISpot plate. So the ELISpot plate is there to capture the signals that the T-cells make. And on the second day, we take the cells off, we add some different antibodies and colours, and at the end of it, you end up with a well with a white background and lots of blue spots in it. And each spot is the footprint of a T-cell that has been produced by the vaccine and is able to respond to the virus in real life in the future should you encounter it. It's always exciting to see lots of spots, and the more spots in the well, the more T-cells your body has made in response to the vaccination. And hopefully, if T-cells are effective against the COVID virus, that means you've got lots of T-cells ready in your body to fight off the infection should you encounter it. We like to look at the immune response over time after vaccination. So we'll look to see 2 weeks after vaccination, which was when we would expect you to have the highest number of T-cells in your blood. But what we're really looking to see is that those T-cells persist for 6 months, 12 months after vaccination, so that hopefully that immune response will last and give you a reasonable period of protection.
So to simplify and analyse it, essentially what we're trying to do is take blood from a vaccinee. We will then separate out the serum, and then we will try and test it to see how much or what level of antibodies are in there that we can measure against SARS-CoV-2. So we have a plate, and in those plates we've coated that with a part of the virus, so a protein from the virus. Then you stick your antibodies on top. If the antibodies recognize the protein, it will bind. You come in with a second antibody to see if you can detect any of the antibodies that have bound the protein or the part of the virus that you're interested in, and then you get a colour change depending on how you're running the assay. We get very excited when we see our plates going from a blank to a yellow or a blank to a blue, depending on the type of assay we're running. We use a machine to read out the optical density that is emitted from detecting the antibodies in each of these wells on the plate. So a higher optical density or a higher colour change means that we've got a stronger or a higher antibody response. So what we like to do is test serum at different time points after vaccination, so we can really build up a vivid picture of how the antibody response changes over time after vaccination.
In addition to ELISpot, another technique for measuring T-cells is flow cytometry. This technique uses a technology that simultaneously measures multiple light-based characteristics of individual immune cells as they flow in a fluid stream through a series of laser beams of differing wavelengths. The characteristics measured include a cell's size, granularity or internal complexity, and fluorescence intensity for cell markers that have been tagged with a fluorochrome, a fluorescent marker. By using combinations of known markers, we can count the populations of different immune cells in our test sample, including those responding specifically to our vaccine. Data from all our assays helps us publish a picture of how the immune system responds to our vaccine. We publish this data along with the safety data from the clinic in papers, such as the Phase 1 paper from the COVID trial shown here. Such publications help inform both the greater scientific community and the public of our progress.
So, vaccine trials normally take a number of years to complete. The early stages, you have to show that the vaccine is safe for the volunteers and produces an immune response, and then in the later trials, you have to show that the vaccine is safe for larger numbers of people and different populations and different age groups, but also show that the vaccine is effective at preventing disease. Ultimately, when all these clinical trials are complete, the data that we've collected from the clinical trials is submitted to a regulatory authority who decide whether the vaccine is ready for approval and roll out into the general population. So this brings us to the end of our tour. Thanks for listening, and I hope you've enjoyed this video. If you'd like to find out some more information about the vaccines being developed by the Jenner Institute and Oxford Vaccine Groups, please check out our websites.